this was all part of the same research that I presented on yesterday. So yeah, if there is any questions for those who missed that, just come and ask me afterwards. So the whole concept behind my thesis was to combine quantitative and qualitative methods, so the traditional methods of review with the more modern data mining methods in signal detection and evaluation in pharmacovigilance. So in this presentation, I'll just give a bit of the background of the overall research. The method involved in using those weightings that we came up with yesterday through the Delphi method, the results of applying those to PRR calculations for disproportionality, the conclusion, and some ideas for further research. So I'm sure everybody has seen this picture before, and as it was mentioned yesterday a few times, the thalidomide disaster from the 1960s. But I think it's important to remember that this is really where PV methods started, and we've come a long way, but there's still a lot more room for improvement. So in an ideal world, prior to marketing, we would have all the pieces of the puzzle, all the information we need to say what would be the right dose for the right patient at the right time and what their risks would be in taking that product. But unfortunately, we don't have all this. So spontaneous reporting data is the largest part of information, or the information we can get from spontaneous reporting is the largest and contributes a lot to signal detection. And there are many other types of data that can be used to monitor potential safety signals. So in reality, the picture is a little bit more like this. So we have a lot of information missing that we need to gather post-marketing. So the use of statistics in PV are used to compare the observed number of reports to the expected number in the background data. And the background data is only a portion of what we have. So that's just the calculation that was used for this research, the PRR. So as I said, the D circle there is your database, and this is only a fraction of the reactions occurring, and that is only a fraction of the entire population getting the drug. So the statistical weighting, so the important associations that we measured yesterday were based on the cardiac failure SMQ, and the weightings then from this were used to give a higher numerical value to certain terms prior to disproportionality in the hope to augment it and potentially identify signals earlier. So the rationale behind the chosen medical concepts. At the centre of this research was the ability to identify the important risk factors to monitor during initiation of a medicinal product to the market from the information available prior to widespread marketing authorization. So the rationale for choosing cardiac failure and the other terms that I used for this research came from the information available on the EMA website, from the scientific discussions that were submitted, and then marketing authorization was approved based on those. The main issues for current treatments with diabetic patients were concerns about adding to the cardiovascular risk, weight gain, edema, potential association with pancreatitis, and concerns about renal failure and the unknown long-term safety of these products. So just briefly again, SMQs group terms from various SOCs together in MEDRA to aid identification of a medical concept. So the SMQ cardiac failure, which I discussed yesterday, has terms from all of these different SOCs put together. So this was the average rating, so the mean ratings after I'd done the first round and the second round of the Delphi study for certain terms. So it was decided at the beginning of the research to weight the true signals first, and then I could compare when they became a signal as opposed to with the weighting and without the weighting to show if the method was indeed effective. So these were the terms that were weighted first. Also, the crude weighting from the means, if, like, see for congestive failure there, the weighting was 3 overall. So if we apply that weighting with the PRR calculation, one report is automatically a signal, whereas those terms that were closer to 1 are rated closer to 1. If we apply that as a weighting, they're automatically, or like, it's kind of ineffective and doesn't really make any difference. So there was a lot of discussion with Professor Stephen Evans and another statistician at the University of Portsmouth, and they were just said basically it would have to be fine-tuned, and you would have to just apply different weightings and compare your rates of true and false positives and true and false negatives, and go from there. So the first weighting applied was the mean rating, the crude mean rating from the Delphi study, and then I applied the second weighting of the mean multiplied by 0.6 and the mean multiplied 0.7. It was just chosen to start there and see what happened. So applying the weighting, we had two different data sets. The first data set was 2 years, from 2005 to 2007 of yellow. They're both yellow card data. And the PRRs, confidence intervals, and chi-squared were calculated every month because we had the dates that the reports were entered into the database. So this could produce dynamic PRR reports, which was very useful for seeing when terms became a signal. The second year data set we applied for was a 10-year data set from 2000 to 2010, just for the products that we were modelling the method on. And we could also calculate the PRRs, confidence intervals, and chi-squared at the end of the 10 years. Unfortunately, we didn't have the dates that the reports were entered, so we could only do a static PRR report for the full 10-year data set. So this is just an example of one of the static or dynamic PRR reports. So this was non-weighted for pioglitazine and edema. You can see in month 4 there's a slight peak there in the blue line and the purple, so that's when the PRR was over 3 and became a signal, non-weighted. So just for PRR, the null value is 1. So if PRR is 1, then the number of drug event reports for that drug is proportionate to the number in the whole database. And then if it's more than 1, it's disproportionate and therefore becomes a signal. So for pioglitazine and edema, the number of reports was 3, and because the mean weighting for edema was quite high, so the PRR calculations are quite unrealistic, and it's automatically a signal with the 3 reports in the database. So for edema peripheral and pioglitazine in the database, so we can see non-weighted, it was a signal at month 10. With weight 1, then it became a signal at month 9. Weight 2 went the other direction, and it wasn't a signal until month 17. And with weight 3, it was also a signal at month 9. So it was identified as a signal 1 month earlier, retrospectively, in the 2-year data set. And then for the same term with rosiglitazine, in the non-weighted, it was a signal at month 19. And then with weight 1, it became a signal at month 17, and weight 3, it was month 18. So it was earlier, but again, there's some fine-tuning that could be done with the weight. And then weight 2, again, it became later. So in the 10-year data set, just briefly, because we could only do static PRR reports, it didn't show us as much. But edema peripheral wasn't a signal prior to weighting. And then with weighting, it became a signal again with the first weighting and the third. So in the literature now, that was conducted from 2009 onwards, there's many studies that have discussed the association of glitazones, particularly pioglitazine and rosiglitazine, with cardiac failure, fluid retention, and edema peripheral. So it's now considered a true signal for both. And it was a true signal for the non-weighted terms, for edema peripheral non-weighted in the 2-year data set, and it was not a true signal in the 10-year data set, and then became a true signal with weighting. So yeah, that's just some statistics about the rates of these terms with pioglitazine use. So the research aimed to investigate the role of additional data sources in earlier signal detection. And the fair to moderate agreement for the terms, there was fair to moderate agreement for the terms rated is very important, as I showed yesterday. Applying the mean rating had the biggest effect on disproportionality, and all true positive drug event terms were identified with the weighting factors. So none were missed, which is a good sign, but but there's obviously work that can be done to fine-tune it to make sure that we're not getting then too many false positives. So in conclusion, the enhanced detection process may allow more accurate and early prioritization of ADR reports for further investigation and leading to improved proactive signal detection. Some ideas for further research. So rating the terms for more medical concepts by a refined group of experts for each medical area. Application of the weighting factors to more terms for more drugs in larger database and be able to show it over time. And incorporate visual evaluation tools to aid the signal review phase of PV. So there's some examples out there already that can be used with various databases, but a method that includes the weighting for special terms of interest and takes into account many other factors such as age, gender, concurrent conditions, country of occurrence. So co-suspects will aid in evaluation and discovery of trends. And just an idea then as well is if causality could be incorporated into the method. So, I mean, this is only briefly one idea, would be the Bradford Hill criteria to kind of incorporate that into your visual tool. And yeah, then if this was something that we started to work on, but as with most postgrad research, is run out of time and funding. But the idea was to then use this weighting and put together with your disproportionality calculations, whether they be PRR or other, and have a dashboard so you could filter by your active ingredient and the preferred term and come up. I mean, these are just examples, tables that I've pulled from the internet, but the idea would be that you could see everything in one place and make your decision and further evaluation based on this dashboard. Just an idea stage at the minute. So that's it. Is there any questions or comments? Yeah.
I noticed that when you were talking about weighting and you had sort of one event.
Yeah.
So what sort of numbers of events were you talking about in your initial weighting for in this presentation you just given? When you said there was sort of one event.
Yeah. Well, I concentrated down on that because that had the best results for showing when it became a signal earlier, but it did become a signal for 1 month earlier for a few other terms. But so as you've seen yesterday, the SMQ for cardiac failure, we narrowed that down and excluded a couple of terms that were potential duplicates. So we had 69 terms that were weighted, and we took all of those that were true signals at the end of 2010 and weighted all them and seen what happened. So out of all of them, no true signals were missed.
Okay.
But this showed the best results for some of them being identified a couple of months earlier.
So I was thinking that you work with Stephen Evans.
Yeah.
Because in his, if you're looking at the Evans criteria, he's got, you need 3 cases
Yeah.
as a minimum
Yeah.
for it.
It's just to avoid by chance, yeah, and things. Yeah. So, yeah. Yeah, it was very interesting actually getting to meet with him because I met with him quite near the beginning of my research and kind of said the whole concept to him, and he was like, well, obviously the rating scale, if you ask them to weight 0 to 3, you're kind of obviously 0's obsolete and 3's kind of obsolete. So yeah, he just agreed that it was a good starting point, but obviously there's a lot of work to be done then to fine tune it.
There's a follow-up question.
Mm-hmm.
Said that you selected the terms on the SMQ, right? And then you narrowed it down to just what would be identified signals.
Mm-hmm.
Is there a cutoff number of how many events before they can make it to become identified signals?
Yeah, well, the reason I used PRR, sorry, I should have said, was because the data I had was the yellow card data from the MHRA, and they used PRR up until 2006. So it was to allow for a direct comparison. So using the PRR, the number would have been 3 reports, 3 or more, for it to go on. And then they do impact analysis and various other things to see if they need to review it.
And in other cases, are you able to look at more information about the 3 cases that you picked?
I got massive Excel spreadsheets, and obviously all patient identifiers and everything had been anonymized. So I couldn't go back and then further review those cases. but for the purpose of my research was just so the method of identifying the signals and combining the quantitative. So yeah, unfortunately I couldn't, but that was an ethics issue with the MHRA. So
Yeah, because I was wondering whether you could further screen it
yeah,
by applying, like, let's say, other causes of, in this case, cardiac failure, if you want to
yeah.
rule out. And
Yeah, it would have been ideal to have all their patient history and everything, but I give all my results back to the MHRA and they just agreed because it didn't identify any new signals that they hadn't already had or any potentially new signals. So they were happy, and they'd done, obviously this was all retrospective, so they've done their signal detection on this from before 2010 and done their review and their decision. So.
And in your table where you said mean and then weight 1, weight 2,
Yeah.
is that based on the second group?
Yeah, that's the mean from the second round of the Delphi study, yeah.
Do you have any question? Because weighting 2 and weighting 3,
Mm-hmm.
there was only 0.1 of a decimal
Yeah.
between them, but yet there was a significant difference
Yeah.
in when a signal would have been detected.
Yeah.
One went up from something to 27 and the other one was back down
Yeah,
to
it went,
18.
yeah.
Is there do you have any ideas as to?
I think you just have to go even finer than going by 1 decimal point. But the next plan then would be go from either side of your decimal points and try and work your way in and see what would be the best.
When you
But-
did it on the MHRA data, the 10-year data, what sort of volume were you talking about in terms of? I'm sorry, I'm being very specific.
No, it's fine. I can't even remember numbers, but it was back in the day when Excel had a limit and it was more than could fit on one Excel. So it was a lot of work. I mean, months went into cleaning that data and getting it in a format that I could then enter the PRR calculations on your chi-squared. So yeah, it was a lot of work, and it was quite disappointing then that the 10-year data set could only be done and only have your 1 PRR calculation at the end, because so much had to go into cleaning it and getting it in the right format. It would have been ideal to see it month or even yearly would have been ideal.
Because
But
your chi-squared values are quite high
yeah.
compared to, if you look at, because you know how signal detections move forward and PRRs.
Yeah, it's kind of, yeah, it's a very crude method now. Yeah.
Because that's what the MHRA
Yeah.
used.
Yeah.
But when you're looking now these days, they tend to use Bayesian statistics.
Yeah, there's a lot more that can be done in like the
Yes.
shrinkage type of methods.
Shrinkage and
Yeah,
stuff.
yeah.
That becomes really, it's a bottom line.
Well,
Data
that
analytics.
and I was working with Excel, so yeah.
But that's interesting research.
Thank you. Yes, Sandra.
Will this method work on your company's database?
Potentially, yeah. It's something that needs to be investigated at the minute because I haven't been there that long yet, but I've had some discussions. I need to find out a bit more about the background data, and because we're having a lot of data migration going on at the minute, so we need to kind of wait and see what our data set's going to be. But I mean, even if it was to be applied, it was something that would have to be reviewed on 6-month or annual basis, and yeah, the weightings changed compared to your background data, so there's a lot to kind of consider before it would be go live as such. Okay. Thank you.